Welcome, everyone, and thank you for attending today's webinar. I'm Shelley Frisbee, Cancer Commons CFO, and I'm excited to share with you today the presentation, Breast Cancer Treatment Trends for 2022. This is the first in the Pat Looney Educational Series for Client Empowerment, made possible by a grant from the Looney family. We have many of our clients, caregivers, and partners on the webinar today. For those of you who are new to Cancer Commons, we are a nonprofit founded in 2012 and deeply committed to continuous learning and open knowledge sharing. Our team of navigators, patient specialists, PhD scientists, oncology experts, provide compassionate support and assistance to our clients who are advanced cancer patients and caregivers. Our focus is on helping cancer patients better understand their disease and access the best available diagnostic and treatment options. I have a few logistical details before we start. Today's presentation will be divided into 4 sections. There will be an opportunity for live Q&A after each section and at the end of the program. Please submit your questions via the question and answer feature at any time throughout the webinar. Only the panelists will see your questions. A recording of the webinar will be available to all registrants. Now I have the pleasure to introduce to you Pat Looney's daughter, Caitlin, who will share the inspiration behind this 4-part series.
Thanks, Shelley. Appreciate that. Hello again. My name is Caitlin Landisberg, and I am so proud of this series, as it was my mom's sincere interest to share Cancer Commons with as many people as she could. Thank you all so much for being here. My mom also had the great fortune of working directly with Dr. Emma Shtivelman, who you'll hear from today. Cancer Commons are my mom with not only a second opinion throughout her treatment plan, but best-in-class knowledge and information to ask smarter questions and take action surrounding her cancer treatment. Having Cancer Commons on mom's team was a meaningful shift in mindset for her, and we will be forever grateful. It is now my great pleasure to introduce Cancer Commons Editor-in-Chief, Dr. George Lundberg, who is also Editor-at-Large for Medscape and former editor-in-chief for the Journal of the American Medical Association, and Cancer Commons Chief Scientist, Dr. Emma Shtivelman, who is both a molecular and cell biologist with extensive experience in cancer biology, and of course, a fierce patient advocate. Dr. George, over to you.
Thank you very much, Caitlin and Shelley, and also a vital thank you to our staff, Shan, Courtney, and Deb, who are off camera. I'm Dr. George Lundberg, as indicated. I'm Editor-in-Chief of Cancer Commons, which I helped Marty Tenenbaum found back in 2010 and 2011 as an initiative that grew into Cancer Commons. I'm a traditionally trained pathologist, physician. I didn't get into this business of precision oncology and molecular medicine until about 10 years ago. On the other hand, our main speaker, Dr. Emma Shtivelman, who has her PhD in molecular biology from the Weizmann Institute in Israel in the 1980s, following which she went to UCSF as a postdoc fellow. She'd been working in this field from the beginning or even before the beginning. And I'm thinking you're going to figure that out as we go on. I don't know anybody who knows more about precision oncology than Emma Shtivelman, frankly, and she didn't know I was going to say that. Her career has spanned academia and industry, as well as now the not-for-profit world. Today, we do spotlight on advanced breast cancer. My first contact with breast cancer was in 1955, when as a pathologist assistant, I did a frozen section on a specimen out of the operating room with the pathologist, and we diagnosed cancer of the breast, following which a radical mastectomy was performed, which back then was the standard for what you did when you got a diagnosis like that. things, well, later that day, we received the whole specimen, which was the entire breast, got a lot of the chest wall, muscle, et cetera, down to bone, and then the fat pad and all of the lymph nodes out into the axilla. That was standard therapy. Fortunately, it's changed a lot. This program is going to be divided into 4 quartiles, as was mentioned, and they're roughly equal in length. The first quartile will be on testing, the second quartile on estrogen receptor positive cancers, the third quartile on HER2, the fourth quartile on triple negative breast cancer. There will be a pause after each. Deb will be receiving your questions, and she'll sift through them and send to Emma and me the ones that she thinks fit best for each of the sections. Context, very briefly. Cancer of the breast is the most frequently diagnosed, potentially lethal cancer in women in the United States, with about 290,000 such diagnoses per year. But the good news is that 250,000 of those, or 85%, won't die of their cancer. Basically, most of them are cured. They're cured by surgery, by radiation, by chemotherapy, etc. This is not a young woman's disease. More than 95% are after the age of 40, and the age range of 75 to 79 is the main one. The diagnosis is made by the pathologist by name, by site of origin, by microscopic patterns, by grade, by stage, and by hormone testing by immunohistochemistry, which leads us to quartile 1. Diagnosis drives treatment. Testing drives diagnosis. After recognition that a patient's breast cancer has recurred locally, has metastasized or otherwise spread, how does testing influence treatment choices? Dr. Shtivelman.
Hello, everyone, and thank you much for the kind word that I just heard about myself. I would like to dedicate my talk to the memory of Pat Looney, who passed away from breast cancer last year, and also thank Mike Looney and Caitlin for initiating and sponsoring this event. So as George outlined, I will be talking about types of breast cancer, but I will start with testing. When I work with cancer patients, I invariably push 2 things. I advocate for more testing to drive the treatment decisions, of course, and I try to promote clinical trial with promising drugs, of course. So let's start with the first slide. First, briefly about the types of breast cancer, because this is very important. The 3 major types outlined here are treated very differently. The most frequent type is estrogen receptor or ER positive, 70 to 80% of all. Estrogen receptor is a protein that, as you can understand from the name, is driven by estrogen, which women's bodies produce at different levels at different ages. But this is the major driver of ER positive breast cancer, and is now for many years is the major target of treatment in this group of cancers. The second group, which accounts for about 20%, is HER2 positive. HER2 is a name of a protein of a different nature than ER, and it's present in these cancers, and it is a major driver of these cancers, and it's a major target. And the third group is the triple negative breast cancer, which is called so because these cancers do not have presence of ER, HER2, and another protein known as progesterone receptor, which I would not be talking about. It's not a major, it's not a target in treatment of breast cancer. There are also a few rare types of breast cancer that we would just not have time to talk about today. And now we will switch to testing. Next slide, please. So there are several types of testing. One mentioned by George, who knows more than anyone about that and other things, is what I called here routine pathology. There is not much routine about it because it's very important in determining the treatment, as George mentioned. It definitely tests for presence of estrogen receptor, progesterone receptor, and HER2. It also looks at the grade of cancer, which is means to evaluate how fast it is growing, and the Ki-67 index. That is a direct means to know how fast are cells proliferating. And that testing is the first to be done with a tissue isolated during biopsy of any type. It could be repeated if cancer progresses and metastasizes to other sites. The second type that I will mention is genetic testing, and that is blood-based, and it helps to determine if women have inherited or germline mutations that predispose to development of breast cancer. It's mostly reserved for triple negative subtype, and there are a number of mutations that are associated with development of TNBC, especially indicated for patients who have a family history of cancer or are very young at the time of diagnosis. Next slide. The other type of testing that is almost routine, mostly reserved for advanced and metastatic cancers, is mutational testing. It is done with tumor tissue, and it's based on next generation sequencing. It identifies mutations that drive cancer, because sometimes there are mutations other than the major drivers that I mentioned, ER and HER2, that are really instrumental in propelling cancer development. And this could be targeted because we are now in the era of targeted drugs that are designed to attack cancers with specific mutations. The mutational testing also includes additional testing, which is, among other things, analysis for presence of markers of potential response to immune drugs, and I will touch on it later. Next slide. The other type of testing that is very important for early stage cancers, especially estrogen receptor ER positive cancers, are predictive. Women with early stage ER positive cancers do not need chemotherapy very much. It's not 30 years ago when everyone was getting chemotherapy. Now it's not happening. The test analyzes presence of certain RNAs and can predict with a high degree of confidence if women need chemotherapy versus endocrine therapy targeting ER receptor. And there are other tests that use to monitor response to chemotherapy, monitor possible recurrence, etc. And from here on, I will jump to the discussion of types of cancer, unless there are questions that we have related to the first part.
Okay, I have a question. I have several questions, actually. You mentioned that there may be a role for NGS testing, next generation sequencing testing now, but it isn't utilized particularly often. How do you see that playing out?
It is now strongly recommended for testing of advanced and metastatic cancers. It's not really done for early cancers. Stage 1, 2 cancers are not tested for mutations. Because one of the main reasons actually is because, as you mentioned, they're largely curable without looking into mutational context.
Yeah, but then again, what we're talking about today is metastatic or advanced cancer. So
Mostly, yeah.
should all of these metastatic, advanced, recurrent breast cancers in the ideal world be subject to the
Yeah,
next generation
yeah,
sequencing?
I strongly support
It's time to do that now.
that it is time to test all women with advanced metastatic cancers.
Okay, which drives the next question, which is what specimen? Because the patients have had their breast cancer taken out, treated in one form or another, and at a later date, it might be 6 months, it might be 6 years.
Yeah.
Here comes the cancer back, and what specimen should be used for the next generation sequencing? The original one, which it hasn't been saved in a form that it can be used, or do we have to get a fresh biopsy now, or both?
For women that are diagnosed de novo with de novo metastatic cancer, which means that they already have metastasis at the presentation, it is fine to test a sample from the primary tumor from breast or from a metastatic tumor, whatever is easier to obtain. Usually breast biopsies are fairly easy. For women who had a primary cancer, which was, say, treated successfully and they recurred years later, of course, it's a metastatic tumor because most of the time tumors metastasize to different organs or to lymph nodes even. Anything could be tested as long as the site is accessible. For example, liver metastases that are one of the most threatening types, they can be done fairly easily with image-guided techniques.
So there could be evolution of the mutational status itself. So a more recent one might be better to guide what kind of targeted therapy may now be possible, right?
Right, not only targeted therapies, but routine pathology on new metastatic lesions is very important because 20 to 30% of metastatic cancers change their major status. The ER positive cancer can lose ER, so therefore they cannot be targeted with endocrine treatments. And sometimes triple negative cancers, rarely, they gain expression of HER2, so they can be treated with HER2 targeting agents, and so on. 20-30% is a significant number. So testing for new metastasis is a must.
Right. Meanwhile, going back to the hormone testing, do you have a pretty fair sense of confidence in the results that are coming out of hospital pathology labs, reference labs, regional labs, national labs about the estrogen testing and the HER testing? Now, I'm not talking about NGS now.
The estrogen testing and HER2 testing is very much well developed. There are standards, there are measures. HER2 testing is a little bit more complicated because there are, I might have time to mention it, there are levels, different levels of expression, and only the so-called high HER2 cancers are amenable to treatment with HER2 agents. But this is changing now. Anyhow, the data are very solid. I've never seen patients where there was reason to doubt the results of pathology testing for ER and HER2 and Ki-67, the grade. It's very well based.
Right. As a card-carrying pathologist in the various organizations, I like to hear that doing a good job on the older stuff. The newer stuff, that's another topic, another time. Let's move to quartile 2. Please tell us about hormones in breast cancer. How important are they in treatment choices?
Okay, this will bring me to the next slide, if possible. So estrogen signaling in ER-positive breast cancer is the major force in driving this cancer. And the history of that, it's kind of interesting because back in 19th century, named by a man by name of Thomas Bitson, who was a military man, a surgeon, and a scientist. He was doing some experiments, and he removed ovaries from lactating rabbits, from breastfeeding rabbits, and they stopped producing milk, which told him that ovaries make some substance that stimulate lactation. From this on, he went on to actually remove ovaries of 3 women that had breast cancer. And amazingly, they did much better and lived longer because of obviously the survival of breast cancer in 19th century was short. There were no treatments, period. From that on, it took just a few years to the end of 1929 and 30 when estrogen and actually several compounds, estrogenic compounds, were identified. And then moving on to 1977, the first drug that inhibits estrogen testing was discovered, signaling was discovered, and that drug is tamoxifen. Since then, it's credited for saving lives of probably hundreds of thousands of women, if not more. So tamoxifen, and I start with it because it's the first drug in the series, it inhibits activity of estrogen receptor by simply binding to it and preventing the estrogens binding to ER. Since then, there are new classes and a number of other drugs that inhibit estrogen signaling that were developed. There is an enzyme, aromatase, which is responsible, a critical step in synthesis of estrogen, estron, estradiol. And there are 3 drugs approved that inhibit this enzyme and therefore lower levels of estrogen and prevent activation of ER that drives breast cancer. And there are also drugs known as estrogen receptor or ER degraders, SERD. There is only one approved, known as Faslodex. It, as the name suggests, it simply degrades ER. There are also other drugs, but I may or may not get to them later. And of course, there are other hormonal ways to prevent production of estrogen in ovaries, which is very high in premenopausal women. The old way would be to surgically remove ovaries, but there are drugs, several drugs known as gonadotropin-releasing hormone agonists that, in a rather complicated way, prevent production of estrogen. So, as I mentioned, the three types of drug, aromatase inhibitors, tamoxifen, and Faslodex, are used to inhibit ER signaling, and they've been in use for a while. But the big change happened about 6 years ago when the first targeted drug was approved. It was realized that one of the ways activated ER drives proliferation of breast cancer cells is by activating a certain cellular pathway that leads to cell proliferation. And the key enzymes in this process are CDK molecules. Those are proteins that have activity that leads directly to DNA replication and cell division. Because CDKs are cyclin-dependent kinases, because there are little machines that have a certain function, they could be inhibited. And the drugs are known as CDK4/6 inhibitors. The first one approved was palbociclib. And it works just so much better than estrogen inhibiting drugs alone. It prolongs, it could be used with aromatase inhibitor, mostly letrozole, and it could be used with Faslodex in women who already received aromatase inhibitor and failed to respond to it. I think we can remove this slide, by the way. Thank you. So CDK the introduction of CDK4/6 inhibitors changed a lot of things in treatment of ER-positive cancers. So women now can get, depending on their age, they can get tamoxifen, they can get aromatase inhibitors, first line of treatment. I mean, that's used to be. Now they get aromatase inhibitor with a CDK inhibitor, palbociclib, ribociclib, or abemaciclib, and the progression-free survival is much longer. And I'm talking about metastatic breast cancers. However, one of the CDK inhibitors, abemaciclib, showed a very good activity in women whose cancer was still early stage, but had at least 4 lymph nodes involved. They received neoadjuvant treatment, the surgery, and they actually benefit when abemaciclib is added to the standard endocrine treatment for them for maintenance. It really prolongs the survival and prevents recurrence of these cancers. So with that aside, nevertheless, the resistance develops even to combination of CDK inhibitors and estrogen receptor signaling. This changed when it was realized that about 40% of ER-positive breast cancers have mutations in another protein that is also an enzyme, and it's phosphatidylinositol kinase 3, or PI3K. This could be inhibited with certain drugs. There are probably 7-8 drugs that inhibit that protein PI3K in trials. One of them is approved. It's known as alpelisib or Piqray, and it can be used successfully after failure of CDK inhibitor. So that really expands options for metastatic breast cancer patients, and the treatments could be used sequentially, which is great because women can go from one treatment to another to the third, and then what? Then there are new drugs of different kinds. I will only mention the SERDs. It's selective estrogen receptor degraders like Faslodex. Faslodex is a good, strong drug, but now there are drugs in trials. There are probably 6, 7, and 3 of them are promising because they're probably better based on preliminary results. They work better than Faslodex. So, and they could be given after Faslodex option was exhausted. They could be given again with CDK4 inhibitors, and remarkably, they also work in the context of estrogen receptor mutation. So cancers, ER-positive cancers that are treated with a number of inhibitors of ER signaling tend to develop mutations in the ER that makes it unresponsive to these drugs. And the new SERDs, the estrogen receptor degraders, work in the context of mutated ER, which is great. Faslodex also works a bit in the context of mutations, but these new drugs with long names like elacestrant, amcenestrant, and one with a luckily short name of ARV-471 are the new treatment options available only in trials. So what does it all mean? That means that even not only in early breast cancer, but in metastatic breast cancer, there is an option of going without chemotherapy for a very long time, for years. There are women who've been diagnosed 10, 15 years ago, and they're doing well on treatment that involve endocrine drugs. Once this fails, the option is, of course, clinical trial with new drugs or chemotherapy with usually with one drug. It depends on severity and aggressiveness of metastatic spread. So again, 20, 30 years ago, 90% of women with any kind actually of breast cancer were treated with chemotherapy invariably. And now in early breast cancer, maybe only less than 20% of women get chemotherapy. The other remark that I'll make is that overall survivals in ER-positive cancer has increased from about 2 years or less in 1970 to over 5 years in 2021. Obviously, this includes metastatic ER-positive breast cancers. And now we can take questions.
Okay, here's a question that's a good one. You mentioned ER positive status. What's the significance of progesterone status, positive or negative?
There were multiple attempts to attempt and inhibit progesterone signaling, but they didn't produce any good results. And there are very few, if any, trials targeting progesterone. If anything, some cancers express the androgen receptor, which is a male hormone, and there are renewed attempts to target it.
Okay, one more question from an audience before we move to quartile 3. I realize you didn't mention CHEK2 mutation, but I'm curious if aromatase inhibitors are a standard treatment with ER positive more than 95%, PR positive more than 95%, HER2 with CHEK2 mutation. Any comments?
Yeah, CHEK2 mutation is one of these mutations in the homologous DNA repair pathway, same pathway as is governed partially by BRCA1 and 2, the best known mutations. And not all cancers with this mutation, especially BRCA2 and CHEK2, then they are not always triple negative. They express ER quite often. So yeah, they are and could be and are treated with inhibitors with hormonal drugs.
Okay,
But there are also drugs that target, there are drugs that target CHEK2 mutation.
Right. In the interest of time, I think we need to move to quartile 3.
Yeah.
Tell us about HER2. What is it and why is it important for therapy?
So HER2 is a major target in the cancers that express it. The gene was discovered in 1995. The protein was described in 96 in Genentech, and there were several prominent oncologists, in particular Dr. Slayman of UCLA, who really pushed Genentech for development of a drug targeting HER2 because he was convinced based on the available data that that's a major driver of HER2 positive cancer, and he was right. Genentech put an enormous effort into developing an antibody. So antibody is an immune system tool or component that binds to certain proteins that it recognizes and inhibits, takes them out of the action. The antibody, the name of it is probably well known to most of the listeners. It's trastuzumab, also known as Herceptin. It was very difficult to make a humanized antibody. The antibodies could be derived easily from mice and rabbits, but they cannot be used in humans. So it was a huge effort just to make enough of Herceptin. Genentech ran first trial with just 15 patients. Amazingly enough, some of them responded to treatment very well, and that became well known, and women organized to stage protest and demand compassionate access to this drug, which was actually granted. Anyhow, the drug Herceptin was approved in 98 for treatment of metastatic HER2 positive breast cancers in conjunction with the chemotherapy drug docetaxel, a taxane drug. Genentech also later developed another antibody to HER2 that on its own doesn't have activity. It's known as Perjeta, but together with Herceptin, it works very well. So that is approved in combination. And this is now a standard first line of treatment in HER2 positive metastatic breast cancer. Herceptin, Perjeta, and Ataxan. That actually produces very good responses, deep responses, but eventually fails, like most of the treatment in cancer. And there is another antibody known as Kadcyla that is an antibody of a different type. It's an antibody drug conjugate. So it has chemotherapy drug attached to it. So it's in one molecule combination of antibody and chemotherapy. That is approved as a second line of treatment. There are a lot of other, not a lot, but a fair number of antibodies to HER2 of different types. And the most advanced and approved about a year and a half ago for treatment as a third line is Enhertu. So this is the drug about which oncologists say words like amazing and magnificent and so on. And just to illustrate why it is so highly regarded, I will ask for the next slide. So this is the result of a clinical trial known as Destiny Breast 03 that compared the new antibody drug conjugate Enhertu. Here it's labeled as TDXD. Compared it to TDM, which is Kadcyla. So the blue line is the new drug Enhertu, and the red line is Kadcyla. And the graph shows progression-free survival. How many women did not experience progression when treated with this or the other drug? And you can see that 1 year after treatment is started, more than 80% of women who received the new drug Enhertu have no progression of their cancer versus less than 40 with the old drug, Kadcyla. And that difference continues for a long time, well over 2 years. So it's not very often that you see results of randomized trial like that, and that explains the excitement over Enhertu. I will also briefly mention that Enhertu works in HER2 positive cancers that have low levels of HER2. These are not amenable to treatment with Herceptin or Kadcyla or anything else. They are considered to be HER2 positive, but not anymore. The Destiny Breast 04 trial showed that 60 to 70% of women who receive Enhertu and have HER2 low cancers respond to this drug. And that's pretty amazing. That's a big breakthrough. Not only that, Enhertu has activity in brain metastases, which are unfortunately quite frequent in HER2 positive cancers. There are other drug combinations approved, but Enhertu seems to be just amazing. So with these new drugs, of course, Enhertu, it's approved as third line, but there are not many data. But anyhow, the survival rate for all stages of HER2 positive cancer in the first 5 years is 90%. It's only 30% for metastatic, but I expect this to improve with the development of and use of Enhertu and a number of other drugs for HER2. So I'll stop here for questions.
Okay. Here's a question. You're a staunch advocate of clinical trials. How does a patient with advanced breast cancer search for and find the best clinical trials for that patient?
It's pretty difficult. When I search for clinical trials for every particular patient, I look most of all, not only at eligibility, which is, it could be difficult, or geographical location, but first of all, I look for the drugs that have preliminary evidence of activity. It's very rare that I suggest phase 1 trials with new drugs that have no evidence of activity. So that's all I can say. But my pet peeve, it's very difficult to enroll in clinical trials because I can suggest 10 trials to a patient, and the patient will contact them one by one, and they will reject for different reasons. Eligibility based on lines of treatment, certain other health conditions, the list is very long. But the first step has to be done. Find clinical trials with promising drugs that hopefully have some evidence of activity.
Okay, this goes back a bit, but it's still applicable. You mentioned liquid biopsy, and
Yeah.
there was one question that came in that said, do you get more good information from tissue or from blood testing? And before you answer that one, perhaps you could make it clear to our viewers the difference between cfDNA, ctDNA, CTC. Which ones of them apply the best to advanced breast cancer?
Okay, first I'll address the difference between liquid biopsy, analysis of liquid biopsies and solid biopsies. So there are differences. Some types of cancer, for example, brain cancer, do not release CT, which is tumor DNA, cell tumor DNA. They don't release it in blood. So CT, liquid biopsies are not very useful. In breast cancer, ctDNA is very useful. It's now used, it's so sensitive nowadays that it's even used to monitor a response to treatment in early breast cancer. So it's a great tool because it doesn't require invasive biopsies. The other advantage of liquid biopsy in metastatic cancer, that when testing is done with tissue biopsy, for example, from a lung lesion, but patient has bone metastasis and liver metastasis and lymph nodes, it doesn't reflect what's happening in other sites. They may well have different mutations. Liquid biopsy covers everything that's released into blood. There is still a bias because lung tumors release more ctDNA into blood than, say, lymph nodes or bone tumors. But the best, of course, to do both, especially if there is a new metastatic lesion, it's better to sample it and test, but it's not always possible. The second question was about ctDNA versus cfDNA. This is pretty much the same because the test looks for DNA that floats in the blood, and it's called cell tumor DNA or cell-free DNA. There is also a test for circulating tumor cells, which is much less sensitive and difficult and requires more blood.
Okay, sort of a basic question, but some of our listeners might like clarification. The differences between adjuvant and neoadjuvant therapy, and how does one figure out which would be best, if either?
So neoadjuvant therapy is performed before surgery for early stage cancers. The goal of it is to obviously reduce the size of tumor, or better, to eradicate it entirely, which is known as pathologic complete response. It is not routinely done for, say, ER positive tumors that are small because they can be removed. It is done when there is presence of lymph node involvement, which is tested in imaging studies, ultrasound or MRI, and of course CT scans, less sensitive. And it's called neoadjuvant or pre-surgery. The adjuvant is given after surgery in both cases when there is no tumor left in breast or lymph nodes have been eliminated by neoadjuvant treatment and surgery, women need adjuvant therapy. It could be chemotherapy, and it was back then, but now it's very often just endocrine therapy. In case of HER2 positive cancer, the standard is adjuvant Herceptin, and so on. I hope it answers the question.
Okay, let's go to quartile 4. We're pretty much right on time.
Yeah.
Quartile
So-
4 is, what is triple negative and why is it so bad?
In my opinion, triple negative is so bad because it doesn't have a clear driver. It doesn't have a clear target. It's kind of bland cancer in these regards. A certain percent of TNBC is driven by BRCA1 mutations and some other mutations in the same pathway of DNA repair. The women with these mutations have a far higher risk of developing breast and ovarian cancer. Chemotherapy always was the major modality for treatment, and it still is, with one significant difference. Immune drug Keytruda is now approved for treatment of metastatic TNBC in first line. It's combined with chemotherapy. It prolongs overall survival by 7 months. Unfortunately, to qualify for the treatments, women's cancers have to express a protein known as PD-L1. This is a marker of response to Keytruda, and not too many TNBCs have this marker. So for women with PD-L1 positive cancer, that's a good option. For PD-L1 negative, it's not applicable. Keytruda is also approved in neoadjuvant, pre-surgery treatment for early stage TNBC. Other than that, the drug that's not really chemotherapy, it's an antibody drug conjugate that targets a protein known as TROP2. That protein is present in cancer cells that divide the fastest. So it's a pretty good target. And the antibody drug is known as Trodelvy. It is approved as a second line of treatment in TNBC in women who failed chemotherapy or chemoimmunotherapy. It improves overall survival from 7 months to 11 months. So it's active, but not terribly so. And there are new antibody drug conjugates in trial, notably one known as Dato-DXd that seems to be better than Trodelvy. I think I should stop here. We have 5 minutes left for questions.
Okay, here's some additional questions have come in from our audience. Since Her2 results are so favorable, do you see it being prescribed for either first or second line therapy?
It is now under approval by FDA, under review by FDA for approval as second line therapy.
Okay, so the
It will
answer is
probably
yes.
happen soon, yeah.
Yes, for second line, good. Are you able to make any comments about the Her2 vaccine trial?
Not really, because I haven't seen results, but I would say what I know about targeting HER2, I didn't mention the very new approaches that involve CAR T cells. Those are patients on T cells modified to recognize HER2. There have been some successes with that. And also NK, natural killer cells. It's another component of immune system that also could be modified to recognize the certain proteins, HER2 in this case. This also seems to be promising.
Are you a fan of accelerated approval? Do you think the FDA's program is working well or might need to be revisited?
Folks, usually accelerated approval relies on results seen with PFS, progression-free survival. It's not how long a patients will live after this particular new treatment, but how long the patient will not experience progression during this treatment. And drugs get approved when PFS is improved by 2-3 months, which is, it's not acceptable. On the other hand, it's difficult, I guess, to say no for some extremely serious cancer where overall survival is an year or so. So it's a difficult question. Of course, PFS as a surrogate marker is not the best one.
It seems to me like if you look back before there was accelerated approval, and then you look now, it's better that it does exist. I think safeguards are there up to some extent and allows promising drugs to make it through. Okay, here's another one from a viewer. Do drugs like Trodelvy cross the blood-brain barrier?
Not very well. There are not much data on the activity of Trodelvy but in crossing blood-brain barrier. That's why everyone was surprised when in HER2 for HER2 positive cancer was able to do it, because it's a big molecule.
Okay. Did you talk about Keynote 522 and the neoadjuvant pembrolizumab in triple negative?
I mentioned it very briefly, that it is approved based on results of Keynote 522.
Okay.
And it increases complete response in the primary tumor by about 15%.
On a practical level, this patient says, I use an EMR to follow my diseases. How well do they work with following metastatic breast cancer?
EMR meaning electronic medical records?
Electronic medical record. How well do they match up with what they offer the doctor, the nurse, everybody else to put in, and what the patient can now see, since obviously the medical record belongs to the patient?
Yeah.
It has a right to follow. But how do you have experience with how that works?
I look at records every day, and I'm very happy to see if the oncologist provided detailed notes, including the potential future treatment options. It doesn't happen too often, unfortunately. That's all I can say. They're very good in summarizing current treatment, responses, side effects, but future is hardly addressed in EMRs.
Okay, well, if you had to take a choice right now in metastatic or advanced breast cancer, bet in the future, targeted therapy, immunotherapy, or something else is going to be the best?
I would think that there is still future for immunotherapy in triple negative, which is good. I still have hopes for that. There is room for targeted therapies in every type of cancer. It just needs to be understood. The mutational context needs to be defined, because there are sometimes mutations in the genes that are not that common. For example, FGFR, there are good drugs and women with breast cancer can get them, but mutations are rare. And the list goes on. So I would say combination, there is still plenty of room for surgery and chemotherapy and so on.
Okay, my guess is that our audience has come to understand why I described you as my favorite precision oncologist at the beginning of the hour. I think maybe they'll get that. They'll begin to get that. So thank you very much, Dr. Emma Shtivelman. Keep up the good work, and thank everybody else. And let's kick this back to our COO, Shelley.
Thank you, Emma and George, for a wonderful and informative presentation. And thank you all for attending and participating today. We will be sharing this recording with everybody who is registered, and we'll be posting it to Cancer Commons website and YouTube channel. Please send any additional questions you have to education@cancercommons.org. If you or a loved one need assistance managing advanced cancer, Cancer Commons is here to help. contact us via our website, cancercommons.org. And finally, Cancer Commons is extremely grateful to the Looney family for sponsoring this series. Our next webinar will be held May 18th and will focus on issues relating to caregiving and caregiving needs. We look forward to having you join us. Thank you again.